Feral Pathogens. So it is my absolute pleasure to introduce the plenary speaker. It's Dr. Geoff Pegg. He's a Senior Principal Forest Pathologist and Team Leader Forest Production and Protection with the Department of Agriculture and Fisheries in Queensland. Prior to this, Dr. Pegg worked as a Biosecurity Officer with Australian Quarantine. Dr. Pegg has more than 24 years' experience as a forest pathologist working on a range of diseases impacting commercial forest species. For the last 14 years, following the arrival of Ostropochynia cityi, myrtle rust, Dr. Pegg has focused his research towards the environmental space, studying the impacts of myrtle rust on native Myrtaceae and associated ecosystems. Dr. Pegg has been working in partnership with Indigenous groups to develop and deliver projects to address exotic pests that threaten the cultural and environmental biodiversity values unique to Australia. This includes myrtle rust and the more recent decline of bunya pines in the Bunya Mountains National Park. This morning, Geoff's presentation is Myrtle Rust in Queensland, Current and Future Challenges in Managing the Impacts. And it's my pleasure to welcome Geoff. Thank you.
Thank you, Ange, and good morning to everyone. Firstly, I'd like to acknowledge the traditional owners of the land on which we're meeting today, but all those that I've managed to work with or had the privilege of working with in my myrtle rust journey, particularly, I guess, the Butchulla Land and Sea Rangers, where I've spent the last 6 or 7 years working with them and continue to work with them in the environmental biosecurity space. So just a really brief introduction, I guess, to this topic. I think the title, Bob might have actually dreamt up the title, but so the word challenges. I mean, from a plant pathology perspective, we're used to dealing with it could be a pathogen that has multiple hosts, but we're usually just dealing with a single host at a time. So we got really detailed knowledge, I guess, of the pathogen, the host, and the interaction in our ability to try and develop a management strategy. In this case, we're dealing with a pathogen that's got a huge host range. It occurs in multiple different ecosystems. There's all these different factors influencing the disease. Then we get on top of that massive fires that influence the disease. We get floods, cyclones, etc. So it's a really complex system and one that I find really challenging as a pathologist in trying to manage. So I just want to go through first, I guess, I'm talking about some of the challenges. I have to go through, I guess, some of the history of what's occurred. So early, I guess, in the 1980s, it was identified impacting eucalypts in Brazil. So it first came to our attention as being a significant threat to Australia. There was additional work done in terms of looking at host ranges, work done by CSIRO showing it wasn't just eucalypts, that there were a lot of native species that were at potential risk from this pathogen. So a lot of processes were put in place in an attempt to try and reduce the risk of myrtle rust, or back then known as guava rust or eucalyptus rust, coming into Australia. Then in 2004, viable spores were detected on a consignment of timber, eucalypt timber, coming from Brazil, and that pathway was shut down completely. Nursery stock, 2 years in quarantine minimum, so you couldn't bring in any Myrtaceae without going through strict quarantine processes. Then they developed a contingency plan. Unfortunately, that's basically sat on someone's desk and collected dust, even when we had the detection in 2010. And I won't go into the politics, I'm sure Bob will cover a lot of that in his talk after me. Soon after that initial detection in New South Wales, it arrived in Queensland just before we had the Brisbane floods. Again, it was nursery stock that was the primary source of spread, moving up and down the coast. But by 2012, we saw rust was pretty much up and down the east coast of Australia, so in Victoria, right up to Cairns. Initially, again, nursery stock, but we also started to see a lot of natural spread occurring as well through windborne. So for those who don't know rust, it's really easily spread. It's a windborne pathogen. By 2017, we started to see the first species really declining. The first 2 species were listed as critically endangered, so the native guava and the scrub turpentine. And since then, we've had additional species. We've seen more and more species declining. And we're seeing more and more things that we didn't expect to decline, starting to decline now. In 2020, the Myrtle Rust National Action Plan was published, but it hasn't been adopted at a state or federal level at this point in time. The disease continues to spread in 2022 with the first detection in northwest WA. So it's still making its way around into different ecosystems, to different environments affecting different species. So this one, and I can talk publicly about this one, it is public information. So an exotic strain was detected using eDNA, and this was in Sydney. So eDNA, if we look at that from a biosecurity perspective, it's a bit of a grey area. There's no real response that actually happens based on a detection of eDNA. But I asked the question, I suppose, if it was eDNA of something else like Xylella or one of these other agricultural important species, would the response that we got with rust, in this case, be the same if it was Xylella? What happened was there was a really we're not quite sure what to do. Some additional surveys were done by Angus Carnegie. We can't find any trace of that exotic strain. So where did this come from? We still don't know. So let's look back at what's happened over time, and this is more Queensland focused, I suppose, more than anything. So when it arrived in Queensland for the first 4 years, we found 61 new host records added to the global list. 48 species we considered as highly or extremely susceptible, and this was based on leaf symptoms only. This is not on dieback levels, and it was based on a system that we adopted from Hawaii and Brazil in terms of assessments. So those 48 species, they did include species that had a range of susceptibilities. For example, some of the paperbarks where we've seen good levels of resistance, but we're also seeing high levels of susceptibility. But it also included species that we saw in botanic gardens, but wouldn't necessarily be in the native range of where rust does occur along the east coast, so further inland, west of the dividing range. But it did also include a number of species, or a high level of species, that we're now seeing in severe decline. So then we started looking at particular species, using those susceptibilities. We targeted particular species, and it was more of less a political angle when we first started these surveys with Rhodomota sidioides and Rhodamnia rubescens, to try and say to the environment departments, there is a problem here, because it was all focused around industry. But we had to say, well, environment, you need to pay attention to what's going on. So then in 2016 we demonstrated there was a dramatic decline, and then there was additional surveys done with Fensham et al. In 2018 of those two species, or the Rhodamnia Rhodomota sidioides. And then, as I said, there's been continued decline in species, with a further 16 rainforest species identified as in significant decline. There's not much in the way of studies that have been done from a plant community level. A lot of it's been focused on a tree-by-tree basis, looking at tree decline or species decline. We've only got 2 sites, that we've looked at broader ecological impacts. The first study that I was involved in, this is in a Myrtaceae-rich wet sclerophyll system down in southeast Queensland in the Tallebudgera Valley. Again, I was more focused on looking at species decline within these ecosystems rather than the flow-on effects. But we saw there that we were looking at more than 3,000 trees per hectare dead or dying within this system while we were doing those assessments. And we saw a dramatic decline of a range of those species. Some of those were early colonisers and young trees, so they're regenerating within this site, killed within the study period that we were conducting, but we also saw some of the overstory rainforest species like the Syzygium coriolanthum starting to decline, and some actually dying, so 80, 100-year-old trees being affected by rust as well. So Christy Stevenson did more of a broader flow-on impact study as part of her PhD, and she's talking later today, showing that there was a big change or shift in this environment as a result of rust, that we got a shift to non-Myrtaceae rather than Myrtaceae regenerating within the seedling populations. And it was also demonstrated that the fruit size that was coming through and replacing those original fruit was going to be smaller, and there was going to be a period where we're likely to get a short-term reduction in food availability and local bird visitation rates. So the flow-on impacts were there. How significant they are, we're still working on that space. The other area that's been looked at from an ecological perspective is the keystone species, the water gum, or Tristaniopsis exiliflora, by Meikle-John et al. In 2021, where there was a significant dieback, particularly in the wet tropics along all the waterways there, where the loss of this particular species could have significant flow-on effects to the Great Barrier Reef, with destabilisation of the banks and potential sediment runoff into these ecosystems. Also, I guess there's the direct impact in terms of those ecosystems themselves, in terms of increased light levels entering these sites and increased water temperature as well. And I'm not going to go into great detail, and Christy's going to talk about this in a minute, but there is a follow-up project on both these ecosystems. This project's split up into two sub-projects, one in the Gondwana rainforest areas and those surrounding ecosystem, and the other one is an extension of the Kanuka box that's being led by Darren Crane. And so it's looking at those broader ecological impacts of the loss of Kanuka box within these ecosystems. But it's also looking at the genetics of the populations across its distribution. So it's genetic variability across its range and also within its range to see what we can do from a breeding perspective or reintroduction perspective if necessary. We haven't done too much in this space. It's something we're trying to work on, but as I said, I've been working very closely with Butchulla Land and Sea Rangers, trying to get an understanding, I guess, of not only the cultural impacts, but how we would look at managing myrtle rust from a cultural perspective, rather than just the traditional ways that we look at in terms of collecting germplasm, storing germplasm, doing the testing and that sort of thing. So our surveys were fairly thorough, I guess, across Gari, as thorough as they can be, because there's big areas that are pretty inaccessible. But 11 of the 26 culturally important species were identified as susceptible. Varying levels of susceptibility and varying levels of impact were identified. A lot of the impacts we saw post-fire too, so things like the syncarpia or the satinae, we didn't see any impact until post-wildfire. Fortunately, we've got additional funding now through Federal DAF to not only look further at this work on Gari, but actually extending out to other Indigenous ranger groups. So that project's been led by AJ Perkins, a Gumbungia man down in Coffs Harbour. So as I said right at the start, there are many, many challenges we face. You know, the number of different hosts, the number of different ecosystems being impacted. So where do we start? Obviously, from a species perspective, you know, based on that species level decline, the rates of impacts, that's obviously where we're going to start in terms of the listing. We don't have much in the way of structured monitoring programs to see what's happening with other species. Those species that are listed as moderately susceptible or low susceptible, do we really actually understand what the effects of repeated infection are going to have? Do we understand really that even those low levels of infection is going to impact on flowering rates and fruiting rates? I've seen with some species, like some of the Rhodamnia sessiliflora, that an individual that's showing good levels of resistance, for some reason the fruit and flower will get infected. Whether that's across the populations or whether that's individuals, I'm not quite sure. I guess the question from a funding body perspective would be, is the species by species approach sustainable? There's a lot of money and a lot of investment goes into obviously collecting the material, then obviously storing the material. And Alice will actually talk a little bit more today, but no pressure in terms of that sort of work that we will require in terms of being able to collect and maintain a lot of these species. So what about looking at these species from an ecological importance perspective? How do we actually rate them in terms of their importance in an ecosystem? A lot of the literature that we've seen on many of these Myrtaceae is just the taxonomic description. It doesn't actually give any information on the ecological significance. Are we just looking at keystone species, or should we just be looking at all species as being ecologically important? I feel at this point in time there's still a lack of clear direction and long-term vision of what we're wanting to achieve. So it's necessary that we're getting together as stakeholders and actually working out what this vision, long-term vision is, so that we can actually get good direction and good collaboration. But I think, and I'm just putting this slide up to basically indicate that the science community has met the challenge. Despite the limited amount of money, or comparatively limited amount of money, we're actually producing a lot of good information. There's a lot of people out there getting little $30,000 projects up, producing a lot of good information. And so there's a big group of people out there. There's over 200 authors that have published. You know, we sit pretty high in terms of the rankings internationally in terms of rust research. So what about the road to recovery? What's it look like? And I don't have all the answers. This is just part of, I guess, a process that we need to go through to find the best way of getting there. So obviously there's the natural process. So things are going to get infected. They're going to actually select out resistance naturally. We see that with some individuals, and in this photo here you can actually see that's the Satnay, where we're getting infected seedlings. They'll get killed off. Those that are disease-free will grow through. Are they resistant? That's another question. The problem with that is what we've seen with eucalypts, when we've done glasshouse screening, is that we select for resistance to rust actually increases susceptibility to some of our endemic pathogens. So does that mean that if we're getting these natural selections for rust occurring, are we shifting the dynamics a little bit in favour of some of these native pathogens? They're all things to consider. Then we've got other opportunities, obviously, that are going to require intensive programs, breeding programs. It's going to require a lot of coordination, multidisciplinary approach, collecting a lot of genetic material, looking at the genetics, looking at the diversity across populations, within populations, employing tree breeding processes, selecting for resistance, and then obviously reintroduction. How do we actually do that? What's that actually look like? It's obviously going to need to have an understanding of the ecology of a lot of these different species to know how we would look at a reintroduction program. Then there's species that are obviously got no or limited genetic diversity and have no resistance within the populations. So breeding is not a financially viable option for a lot of those species. But again, it's a significant investment to collect these, and we don't want these species going extinct. So we look a little bit more closely at breeding for resistance. It's not just about multiplying up individuals that are showing tolerance or surviving in the wild. It's actually, we need all that genetic diversity. We need those highly susceptible ones in there as well to build up a proper genetic base to launch these breeding programs. We need to understand the relatedness to other species. Is hybridisation an acceptable option? I suppose in a forestry perspective, hybridisation is one of the key things we go to in terms of disease options, but is that an environmentally acceptable pathway? The other reason why we need this diversity is because of the pathogen. We've seen in Brazil all the resistance breeding that's happened with eucalypts over there. A number of times it has overcome resistance. They say it's through mutation, or a lot of the publications would suggest that it's through mutation, but they're now starting to think it's actually recombination that's occurring. But it's quite a few times, they're saying every 9 to 10 years, they're finding that their resistance has been overcome by either a different strain or a variation in pathogen. Recombination is occurring in the New Zealand population, which is the pandemic strain that they've got as well as in the South African strain, which is a unique strain. Obviously, recombination results in high genetic diversity. What that means in terms of overcoming host resistance, we don't fully understand. What it means in terms of extending the host range, we don't fully understand. If we have outcrossing with different strains, maybe that's a greater level of genetic diversity that actually would threaten different host species. We haven't completed these studies in Australia. We need to actually do it. We know teliospores, or the sexual state, is quite common here, so it's highly likely that we're seeing change in the pathogen population here. We're not implementing that within any of our glasshouse controlled rust programs because we just don't understand what that level of diversity within the population is. So the importance of collections. Obviously, we want to go out there and collect it before it's too late, before it disappears. As I've said, to assess that genetic diversity so we can have a better understanding of these species within their native ecosystems. And then it's really important for us in terms of actually determining resistance levels within populations. Comparing what we're seeing in the field, if those that are surviving, are they truly resistant? Or is there something else at play? If we look at the disease triangle side of things, there's a lot of factors that will influence whether infection occurs, whether disease is successful within these individuals. So we have to understand whether that's true resistance within those populations. This is just an example of some of the work that's funded through DESI and led by Fiona Giblin and Tracy Menzies, looking at the native guava and the scrub turpentine, where they've across the populations in Queensland, and it mirrors the work that's been done in New South Wales as well. where we've collected samples from across those populations. They've been sent down to RESA for analysis in terms of looking at relatedness within sites and differences across sites as well genetically. So this is the first lot of screening work that we've done with native guava. So this is glasshouse controlled screening. So this is where you're obviously making it perfect conditions for the rust to infect and disease to develop. Unfortunately, the populations that we've looked at so far, so this is only half the material, we're still working in this space. There's no resistance or tolerance that we've seen within the populations. There is some variability maybe in levels of susceptibility. Unfortunately, the Toowoomba, there was an individual plant or a couple of plants there, hardly any rust symptoms on there, fruiting. It's not resistant when we've infected, which is really unfortunate. But it does seem to have a lower level of susceptibility in comparison to others. So there's really important information we need to get from that in terms of looking at, well, why is it maybe less susceptible? And is there a genetic characteristic that we can look at there as well? So this is probably a big question, and this has been posed at various levels, and it goes back to the whole biosecurity space and the industry environment space as to why plant pests and pathogens fall into this crack. Myrtle rust is a perfect example. So post-transition to management, when industry said, no, it's not really a problem for us. The eucalypts wasn't impacting significantly. Nursery could manage it in terms of either getting rid of those susceptible lines or using chemical, even though you'll still see infected material in the nurseries. Government departments lost interest. We were basically told from an agricultural perspective, it's an environment problem, it's not an industry problem. So we've struggled to get our managers to see the value in terms of what we're doing. We haven't had Bob will go into this in greater detail, so I'm not going to harp on it for too long, but we just haven't been able to engage environment space. And more recently, when we thought we were getting down the track in terms of I guess, some progress, they turned around and said, oh, maybe this should go back into a biosecurity space. It's not a biosecurity problem anymore. There's continued responses, there's always new things coming in. That's biosecurity's responsibility. There's just this big grey area. We need to probably sort this out, and how do we actually shift, I guess, the focus of environmental pests and pathogens, and how do we look at it differently? I don't have the answers, but it's a question we need to pose. It's only a matter of time before the next thing arrives. You can see from that graph there, from the late 1800s right through to now, we've had this progressive incursion of different pests and diseases. This includes commercial forests, so it includes pests and pathogens of pine, the exotic pine as well. You can see they're always coming in. Some not so bad, others moderate impacts, but then obviously we've got some that have had significant impacts. We've got things like polyphagous shot hole borer that's spreading around the world and has unfortunately arrived in WA, although it's still undergoing an eradication program. This thing is a big worry. Those who don't know, it's got a massive host range. It's a tiny little borer, and what it does is vectors a fungus, and it's usually a fusarium fungus, and the fusarium kills the trees. So in that photo there is AJ standing just in front of two large ficus trees that had to be removed because they were absolutely smashed, I guess, by, for want of a better term, by polyphagous shot hole borer. So this is a big concern. It's just turned up in Brazil, so that the chance of this occurring is pretty high. We have the T-shot hole borer on the east coast, which is affecting our Cupaniopsis in urban spaces only at this point in time. So this is just another broad question to pose. Should we perhaps consider environment to be an industry? And I say that based on, I guess, the information I've got in that table, where if we look at the two pathogen situations, both situations in North Queensland as well, where you've got the banana industry worth about 600 million per annum, versus from the wet mar information, the wet tropics, $4.4 billion revenue per annum. Two different diseases. Panama, there's been over $40 million invested in terms of research, and that's no problem with that. That's the sort of thing that should occur. But if you look at myrtle rust, in that space in the wet tropics, there's probably been less than $500,000 invested in that space. And yet that's where a lot of those rainforest species that Rod and his team identified as being in significant decline. So this is my last slide, and I just want to say, because we've got so many ecosystems, so many species affected, and it's really, should be a wake-up call in terms of, I guess, the threat that these pests and diseases pose to our ecosystems, that we need to create capacity. We can't just do it as government agencies, we can't do it as individuals. It has to be a community, land manager, all those sort of groups. There has to be engagement with all those different key partners. But to get this done effectively, we need leadership and coordination, and that's whether that's federal level or whether that's state level. It's a key to making sure that this is successful. Thank you.
Thanks so much, Geoff. That was a very interesting presentation, and I can answer a little bit on that with possibly some direction and leadership. That TSO has committed to developing an action plan for Queensland this year for Myrtle to guide a coordinated and collaborative approach and give some direction on how we can go about trying to tackle these issues in the landscape. So I'll be speaking to a lot of the key stakeholders. I'm the person running the project, so I'll be speaking to key stakeholders very shortly and start drafting a plan for Queensland that'll build on the national action plan that Geoff and Bob and Angus developed previously. So does anyone have any questions for, sorry, I'm not sure where the mics are. Oh.
David Coates here, Geoff. I was just interested, what is known about the genetic mechanism of resistance in both the rust and the potential, the various hosts?
Yeah, so there's a little bit of work going on in Maurizio's team, actually, in the Melaleucas, so they've actually progressed that a little bit. I won't go into detail because it's not my sort of area of expertise, but that's some quite interesting work there. From the eucalypt perspective, the work that was done in Brazil, they first identified that it was a specific gene that was controlling it, but since then a lot of the work has shown that it's multiple genes, and it's quite different. It can be different from one seedling to the next in terms of the genes that are controlling resistance. Other species, I don't think we've got to that stage yet, but it is something that's it's one of those things that needs to be worked on. So.
Yeah, well, I guess you're saying that recombination was one of the mechanisms for generating resistance, and if that's the case, then it's got to be multiple genes operating.
Yeah.
I guess you could combine that with mutation, which means you've got a real problem in terms of trying to figure out how you do develop a resistant.
The rust genome, it's one of the largest genomes they've ever come across. So I think there's still some work in that space in terms of effector genes and all that sort of thing.
Yeah.
But nothing's been, I guess, finalised, I suppose, in terms of some of those factors. But some work was done, Manuka, with the leptospermum, I can't think of the Scapari, and they identified even that there was differences between some that showed stem infection only and leaf infection. There was some genetic difference in between those, and so it's really a lot more complicated, I guess, than what they thought it was based on the Brazilian information. So.
Thanks very much.
Thank you. Does anyone one more question? Might have time for one more. Kathy?
Hi Geoff, it's Kathy Isles. Nice to see you in person, Australian Seedbank Partnership. I'm just going to throw something out there that, in light of you talked about the flow-on effects for ecosystems. I mean, we're talking about three World Heritage Areas now. We're talking about the wet tropics, we're talking about Gondwana and Fraser Island. If we want to get some research around those flow-on effects, I think we have to elevate it in terms of we're heading towards World Heritage Endangered listing for these places. That does get government's attention. Look at all the money that they generated in the research around the Great Barrier Reef. So we do, I think, as a plant science community, need to be communicating that that's where we're heading with this in terms of outstanding universal value of those sites. And with Gari and the Butchula people, they're very, very focused on what that means in the long term.
Yep.
So I mean, it sounds political, but it's actually not. It's
Yeah,
about,
and-
it's about the ecology and protecting those
And
things
Angela
long
was,
term.
and I was just talking about it before, that obviously with the Olympics being in Brisbane, that's another political, the problem is obviously I guess that push needs to come from outside government, because government staff pushing that upwards sometimes just doesn't get that traction. But certainly with the Bachelor, I mean, we've just signed a project with them to actually develop a biosecurity strategy for Gari itself. So, and that includes managing existing threats and that sort of thing. So there is a little bit of action in that space, but again, it's just little bit by little bit.